Okay, so in this video, I'm going to go over chapter 13, and we're going to be talking about energy and catabolism. We're done with all of the genetics stuff, and now we're moving into kind of the metabolism of bacteria, how they can get energy essentially. So we're going to just talk about energy in general, and then electron transfer and energy carriers. Then we'll really go into it with, respiration and fermentation. And then at the end, we're going to talk about some like special or unique things that bacteria can do. Okay. So this is just kind of an overview of how bacteria and archaea acquire energy. So again, remember the trophies that we learned about. So chemotrophy, organotrophy, lithotrophy. So all of this kind of summarizes all of it. And yeah, so I just like this because it kind of highlights everything we're going to be talking about in chapter 13, but also a little bit of what we're going to talk about in chapter 14. So it's a good way to kind of organize all of your thoughts. Okay, so starting with chapter 13.1, first of all, energy obviously comes from the sun. So the sun provides all of the energy on Earth. That makes sense. And photosynthesis is what takes that energy and harvests it. And then, of course, heterotrophy is when an animal eats a plant. So the energy from the plant gets transferred to the animal. And then we have decomposition, where that energy is then released as heat. And of course, every time we transfer from photosynthesis to heterotrophy, there's also some heat that is lost as well. So every time we transfer energy, we also lose a little bit of heat. So that's just kind of an overview of where energy is coming from. So um, in bacteria, they're getting their energy specifically from molecules. So this is just an example. So delta G, by the way, is Gibbs free energy, and that's the energy that's released when you break down a specific molecule. So that's that. And if you'll notice, if the oxidant, so the final electron acceptor, if the final electron acceptor is oxygen, then we call that aerobic respiration. And notice that the delta Gs are very high. Now, notice that it's negative, and that's because we're talking about releasing energy, so it's negative. And notice that when oxygen is involved, there's higher delta G versus when oxygen is not involved. So that's anaerobic respiration. There's not as much energy being harvested. So that's just something to think about. Okay, so before we talk about, cell respiration, let's talk about ATP first. So ATP, you probably know, is a molecule that's used to store energy. It's adenosine triphosphate. So there's the adenosine, and then we add 3 phosphates to it. And every time you add a phosphate, we're putting in energy. So it's basically storing energy in the bonds of the phosphate groups. And then something that the book was also mentioning is that magnesium 2 plus is really important for the cell because ATP is a very negatively charged molecule. And so it struggles to be stable. And so magnesium plus water kind of helps stabilize the structure. So that's pretty interesting. Okay, so that's ATP. There are a bunch of other energy carriers in the cell that you've probably heard before. So NAD plus and NADH. So NAD plus is the molecule that can carry high energy electrons. So instead of carrying energy through phosphate bonds, we're going to be carrying energy through just the electrons themselves. So the electrons are carrying the energy. And so NAD plus is what I consider the uncharged version, and then NADH is carrying 2 high energy electrons. So this is basically a dead battery, and this is a charged battery. Okay. And then of course there's FAD and FADH2. Oh, by the way, I forgot to mention, in plants and photosynthetic organisms, instead of using NAD and NADH, they will use NADP and NADPH. So the P just is an extra thing there, so don't really worry about it. But yeah, they're essentially the same thing. Okay. All right. So like I said, FAD and FADH2 is another high energy electron carrier. So FAD can carry high energy electrons. And then when it does carry high energy electrons, it's called FADH2 because when it carries the 2 electrons, it also adds 2 hydrogens to itself. It does carry slightly less energy than NADH. So we usually say that NADH has higher energy than FADH2, but both of them are carrying 2 high energy electrons. Okay, cool. And then the last thing we're really going to talk about just before we talk about respiration is enzymes. So you probably know that an enzyme is a protein that catalyzes chemical reactions. So what does catalyze mean? It means that we're lowering the activation energy. You probably learned this in chemistry. We're basically making it so that reactions happen faster and reactions happen more readily. And so the way that enzymes work is they have a catalytic site, and this is where your substrates will bind, and then the reaction will happen. And so here, I guess we're transferring a phosphate from here to here. And so this enzyme is helping with this transfer. And so without the enzyme, this transfer would probably not happen. I mean, it might happen, but it would be extremely unlikely. So the enzyme kind of grabs both of them and makes sure that the reaction happens. So super important for all of metabolism is all of these enzymes. Okay, 13.3 really was just going over where bacteria get their carbon. So there's a bunch of different sources. Obviously, carbohydrates is the main one, but they can also get their energy and carbon from lipids. Lignin, which is like an aromatic structure, which gets broken down into different aromatic structures. So aromatics have like the ring structure. And then, of course, bacteria can also break down peptides into amino acids, and those amino acids can ultimately be used for energy as well. And notice that almost every single one of these molecules gets broken down and eventually becomes acetyl-CoA. And acetyl-CoA, which goes into the TCA cycle, obviously generates a lot of energy. So all of these molecules eventually get converted into acetyl-CoA. Okay, so let's start with all of the carbohydrates. So the main carbohydrate that bacteria use is, of course, glucose. So we're going to talk about how glucose is metabolized by bacteria. And so the first step, as you probably remember, is that we have to get glucose and convert it to pyruvate. And then that pyruvate becomes acetyl-CoA, which goes into the TCA cycle. So bacteria actually have three ways to convert glucose into pyruvate. So they have the glycolysis pathway, which is the normal pathway. You've probably learned this in, like a general bio class. This is the general glycolysis pathway. And at the end, you get two pyruvates. There's two other pathways. So the ED pathway, Enter-Deuteroff pathway, it can convert glucose into pyruvate, but it can also convert sugar acids. So basically, it's a sugar, but also has an acidic component to it. So basically, it's kind of like glucose, but not exactly. And so this can be used to get sugar acids and also convert it into pyruvate. And then the last one is the PPP, or the pentose phosphate pathway. This one is, again, an alternative pathway, and it's primarily used so that we can go into biosynthesis. So instead of these molecules really being used to make energy, they're used to make other molecules like amino acids or nucleic acids, other things. Okay, so we're going to talk about all three of these pathways. I know it's a lot. And by the way, I really like this diagram because it kind of summarizes everything and shows how they're kind of connected. And then it gives you the products at the end, how many ATPs and ADHs are made. Okay, so glycolysis is also known as the Embden-Meyerhoff-Parnas pathway. Yep, so EMP pathway. So you should probably have learned this. I mean, maybe not in this much detail, but I'm just going to go over it really quick. So starting here, we have glucose. Glucose is going to be converted into glucose 6-phosphate. Basically, it gets phosphorylated, so a phosphate group is added. And when this phosphate group is added, we use ATP. That's where the phosphate comes from. So when we start glycolysis, we actually have to put in energy first. So there's an input of energy before we can get energy out of it. I like to think about it like when you start a car, you have to just there's that little bit of energy that we have to put in to start the car, and then now the car is running. Okay, so glucose is phosphorylated into glucose 6-phosphate, and then it's converted into fructose 6-phosphate, and that fructose 6-phosphate is then phosphorylated again. So now we have a molecule that has 6 carbons, and there's phosphate groups on either end. So it's double phosphorylated. So again, we've contributed 2 ATPs at this point. So we've used an ATP here to phosphorylate it once and another ATP to phosphorylate it a second time. And then it gets converted into this guy, which you don't really need to know, but as you have noticed, now there are only 3 carbons. So 1, 2, 3, instead of 6, right? There's 1, 2, 3, 4, 5, 6 carbons here. Whereas here there's only 3 carbons. Basically what's happening is the whole molecule gets cut in half. And when the molecule gets cut in half, we get this molecule, and then eventually we'll get G3P, which is the most important thing. That's glyceraldehyde 3-phosphate. So you should definitely remember this molecule. And yeah, we have obviously 2 of them, because when you cut a 6-carbon molecule in half, you get 2 3-carbon molecules. And notice that it has a phosphate, and then the other one also has a phosphate. So that's essentially what's happening here. So we started with a glucose molecule, phosphorylated it twice, then split it in half. So now there's 3 carbons and a phosphate, and there's 2 of these. Okay, so that's like step 1, is going from glucose to G3P, which is glyceraldehyde 3-phosphate. Okay, so we're moving here. Glyceraldehyde 3-phosphate is going to be converted a bunch of times, and eventually we get to pyruvate. That's really the most important part, is that G3P eventually gets converted into pyruvate. But as it's getting converted, we are now releasing energy. So the first thing that happens is we produce NADH. So NADH is produced, okay? And remember that we have 2 G3Ps. So this whole sequence happens twice for every 1 glucose molecule, because we produce 2 of these. So imagine this goes once and then goes again. And so we actually make 2 NADHs total. And then as we remove this oh, by the way, when we produce that NADH, we actually also add a phosphate group. So now it's a 3-carbon molecule with 2 phosphate groups. And again, we have 2 of these. So then we remove 1 phosphate group. When we remove 1, we make ATP. And then later on, we remove that 2nd phosphate group and we make another ATP. Notice that these other steps are just a phosphate jumping around. So it doesn't really matter what's happening here. All you really need to know is that when we produce NADH, we stick another phosphate group on, and then we remove both phosphate groups. And when we remove both phosphate groups, we produce ATP. So in total, we make 1 NADH and then 2 ATPs. But remember that this whole thing happens twice, because we have 2 G3Ps. And so in total, we actually make 2 NADHs and 4 ATPs. Now, remember that in the beginning, we had to contribute 2 ATPs. So in reality, we only actually gained 2 ATPs. So even though we made 4 ATPs at the end here, because we had to use up 2, I guess the net amount of money or energy that you created was not created, but harvested was 2 ATPs and 2 NADHs. And that's kind of summarized here. So remember that. 2 ATPs and 2 NADHs, like the net yield or the net stuff that you got out. Okay, so that's regular glycolysis. This is something that you should definitely recognize. So then there's the Entner-Duderoff pathway. Entner. Anyway, so they get glucose, and guess what? It gets converted into glucose 6-phosphate the same exact way as it happens in the regular pathway. So yeah. Now the next part is the interesting part. So we immediately start making NAD. And in this case, instead of making NADH, we make NADPH. Okay, so that's really important, is that we make NADPH in the ED pathway. And that happens very early in the cycle. Notice that we don't make NADH until later on. But here we start making it immediately. Okay, and eventually we get to this molecule called 6P gluconate. Okay, and so that's like the key molecule that you should be recognizing. So gluconates are what you should be seeing. Okay, and then eventually, you don't really need to know all of these steps, but eventually what ends up happening is the gluconate becomes glyceraldehyde 3-phosphate. Awesome. And that glyceraldehyde 3-phosphate will go back into the EMP, the regular pathway, the regular glycolysis pathway. And then it will go through these steps here. So remember how glyceraldehyde 3-phosphate will go into these steps, and then this will happen. And so that's what happens to this glyceraldehyde 3-phosphate. It goes into the regular glycolysis pathway, and it will produce 2 ATP and 1 NADH. Now, the other thing that happens to this molecule is that it becomes pyruvate. So basically, this gluconate molecule gets split into glyceraldehyde 3-phosphate and pyruvate. Now, the pyruvate is already done, and it doesn't produce any more energy, and so that's pretty much it. So what you'll notice with the enter Deuteroff pathway is that we produce one ATP net. Remember that we're always talking about net amount. And so because we used up an ATP here and we only produced two here, the net amount of ATP is only one instead of two. We produce one NADH, and then we produce one NADPH. So really these two are essentially the same as this. So making two NADH is the same as making an NADH and an NADPH, but it's really the ATP, right? So we made two in the regular pathway. We only made one in this ED pathway. Okay. Yeah, so it's an alternative pathway, but it doesn't produce as much ATP. And yeah, and then the key like differences is that we have like an NADPH that's produced, and then the intermediates are like gluconate molecules. So those are different. And notice that we start producing the NADPH early, and at the end we make a pyruvate and a glyceraldehyde 3-phosphate. This guy goes into the regular pathway, and this guy is already done. Okay, last one is the pentose phosphate pathway. This one is also kind of weird. So we get glucose and we convert it into glucose 6-phosphate. Awesome. And then guess what? It basically goes into the gluconate. So it's basically going through the ED pathway. So we're here, it's going to be glucose 6-phosphate and then eventually becomes gluconate. So it's essentially the same steps here that's happening here. Okay. So all of this is essentially the same. All of the stuff in pink is essentially the same as what's happening in the previous pathway that I just said. Okay, and then this gluconate is used to make the NADPH. Okay, cool. So we make NADPH. But we also decarboxylate. So that's the big thing, is that unlike the other two, we lose a carbon here. Okay, we decarboxylate and we produce CO2. So one carbon is lost, essentially. And so because one carbon is lost, we make ribulose. So that's the big thing, is ribulose. And that's why it's called pentose phosphate pathway, because pentose means 5 carbons, not 6. And so we produce a molecule that has only 5 carbons. So that's ribulose 5-phosphate. And then you don't really need to know all of the rest, but essentially this ribulose 5-phosphate becomes a bunch of different carbon molecules, like heptulose, which has 7, fructose, which has 6. So there's like a bunch of erythrose, which has 4 carbons, right? Glyceraldehyde, which has 3 carbons. So we're making a bunch of different carbon-based molecules that have different number of carbons. We have 7, we have 6, 4, 3, and so there's a bunch of different ones. And all of these guys will go into producing amino acids down here, or producing DNA and RNA, or other things. And so that's the importance of the pentose phosphate pathway, is that what we end up making is a bunch of things that can go into creating amino acids, nucleic acids, etc. And yeah, it's not really meant for harvesting energy. I mean, it harvests a little bit of energy, so we make 1 ATP overall and 2 NADPH. So it's kind of the same as the ED pathway, but really the main thing is for biosynthesis. Okay, cool. So those were the 3 pathways. And then of course, bacteria have fermentation pathways. So without oxygen, what ends up happening is the bacteria start building up NADH, and they have to get that out of the cell. And so what they can do is convert NADH back into NAD+ through fermentation. And so there's 2 types of fermentation that bacteria do. So there's lactic acid fermentation, which eventually makes lactic acid. So there's this. And then ethanolic fermentation, which makes ethanol. So those are just 2 examples, but obviously there are many other types of fermentation that bacteria have. These are just the 2 main ones that you've probably learned, but there's like succinic acid, there's acetic acid, formic acid. And so all of this, we kind of just, especially with the succinic acid route, we call this like the mixed acid fermentation route, because you just basically make a bunch of acids. So, yep. All right. So now that we have talked about that, and we have converted our glucose into pyruvate, guess where the pyruvate goes? Into the TCA cycle, which is also called the citric acid cycle, or the Krebs cycle. Anyway, so yeah, you're gonna need to remember what happens here. So remember that what goes into the TCA cycle is acetyl-CoA. So pyruvate gets converted into acetyl-CoA, but a bunch of other things can be converted into acetyl-CoA, like fatty acids, amino acids, phenols, a bunch of things. essentially become acetyl-CoA through different reactions. So the acetyl-CoA goes into the citric acid cycle, and we lose the CoA, so the CoA gets ripped off, but the acetyl-CoA combines with oxaloacetate. So oxaloacetate has 4 carbons, 1, 2, 3, 4. Acetyl-CoA has 2 carbons, so when they come together, we get a 6-carbon molecule. So that's what these little blurbs are here. So they're saying this is a 4-carbon molecule plus a 2, which makes a 6, and that's citrate, which is why it's called the citric acid cycle. Okay, citrate becomes cis-aconitate, which eventually becomes isocitrate. So really what's happening here is just movement, right? They're all still 6-carbon molecules, just a bunch of things are moving around, okay? But really the main steps are down here. So isocitrate becomes alpha ketoglutarate, and when it converts to alpha ketoglutarate, it loses a carbon. So that's the main thing, is that when we lose the carbon, we produce CO2, and it also makes an NADH in this process. So we call it decarboxylation. Okay, so now we're at 5 carbons, and then guess what? We reduce it one more time, so we're now at 4 carbons. We, by the way, add a CoA back onto there. And so again, we lose the CO2, we make an NADH. So these 2 steps are essentially the same. The only difference is this step also adds the CoA back on. Okay, that CoA gets ripped out. Funny, right? We just put the CoA and then we take it right out. But as we are making this conversion, we produce ATP. Okay, so ATP, or sometimes GTP, depending on the microbe. But succinyl-CoA gets converted into succinate, and when this conversion happens, we produce ATP, and we remove the CoA. So kind of weird. We put the CoA there, we take it right out. Okay, cool. So now the rest of the journey, so we've already produced NADH and then another NADH, and we've produced ATP, and we're at a 4-carbon molecule. The rest of the steps really are just to get back to oxaloacetate, because we want to make sure that we can recycle these carbons. If we just stopped here, then this is a waste, right? So we can eventually convert this back into oxaloacetate so that the cycle can continue. And so that's what's happening here. Succinate becomes fumarate, becomes malate, becomes oxaloacetate. And by the way, when we go from succinate to fumarate, we are lucky we get just a little bit of energy out of it. So we produce an FADH2 as well. Boop, boop, boop. And then from malate to oxaloacetate, we actually also produce a little bit of energy, another NADH. So lots of moving around and producing things. So as you can see, we produce a total of 1, 2, 3 NADHs, 1 ATP or GTP, and then 1 FADH2. So that's cool. Alrighty. Some bacteria can do a different variation of the TCA cycle, which we call the glyoxylate bypass. And the bypass goes like this. So when we get to isocitrate, instead of it becoming alpha ketoglutarate, or this guy 2-oxoglutarate, it's the same thing. Instead of converting here, we're going to bypass and produce glyoxylate. And then this glyoxylate can be converted into malate, okay, by adding it to acetyl-CoA. Okay, so acetyl-CoA and glyoxylate together will become, wait, that doesn't make sense. Yes, it will become malate. Okay, so that's the pathway, and then the malate will eventually go back into oxaloacetate, and then the cycle basically just goes here rather than around the whole thing. So it's a bypass because we're using glyoxylate instead. Okay. So the main reason why bacteria will do this is because it skips these 2 steps here, which are the decarboxylation steps. Remember, here we're losing a carbon, and here we're losing a carbon. Now, if the bacteria are very, like, they're starving and they need to save as much carbon as they can, then they'll go through this pathway because they still produce NADH. So it's not as energetically favorable, right? You don't produce the ATP down here, but you still produce NADH, and you get to conserve your carbon. So the isocitrate, which has 4 carbons, just kidding, no, it has 6 carbons, will be converted into glyoxylate, and then added with acetyl-CoA, makes malate. So yeah, you're not losing any carbons in this process, and so you get to produce NADH without really having to lose anything. And so it's a nice way to save carbons. Okay, cool. All right, so then we put the whole picture together. So remember glycolysis, we make the pyruvate. Pyruvate eventually becomes acetyl-CoA, which goes into the TCA cycle. We already know this. And remember that every time we do this, we are making stuff. So here we're making ATP and NADH. Here we're making a little bit of NADH. Here we make a few NADH and a couple ATP, one ATP. Or two, yeah, every time we go around. But anyway, eventually, all of these electron carriers, the NADH, the FADH, all of these guys will go into the electron transport chain, which we'll talk about in chapter 14. And then, of course, because of that, it produces a lot more ATP. So this is just kind of like a preview of what we're going to be going over in chapter 14, but this is how it all comes together. Alrighty, 13.5 goes over the gut microorganisms, and it was a little bit weird, but one example that they had was sometimes bacteria in your gut can produce GABA. And there it is. Okay, so GABA. And GABA happens to be a neurotransmitter, and this neurotransmitter helps regulate things like pain receptors and stuff like that. And they really haven't done any research, too much research on it, but people are like wondering if bacteria are able to regulate like pain in the gut or whatever. So that was just, I guess, interesting. Anyway, bacteria can get glutamic acid and convert it into GABA, and then it gets exported out, and then who knows what it does in the human. Okay. All right. 13.6 goes over aromatics and how bacteria can break them down. So you don't really need to know all of this, but aromatics, remember, have the ring structure. So here's toluene, nitrobenzene, so all of these guys. One of the big things that you need to know is that almost all of them really, will eventually become catechol, okay, which is this molecule. It's 6-carbon ring with 2 OH groups. And this catechol will eventually, at one point, become pyruvate, which can go into the TCA cycle. Or it can go this way, and then become acetyl-CoA, which goes into the TCA cycle. So either way, it'll go this way or this way, and then get broken down in the TCA cycle. So that's cool. All right. And the last thing we're going to talk about in this chapter is syntrophy. Syntrophy basically means that two organisms are living and working together to help with each other's metabolism. And so this is just an example. So one bacteria produces H2 through its metabolism. So here it's breaking down benzoate. And when it breaks down the benzoate, it goes through the electron transfer chain and blah, blah, blah. All this stuff is happening. But eventually it produces hydrogen. That's like the main thing. And this organism releases the hydrogen, and there's another organism nearby that takes this hydrogen in and then uses it for its own electron transport. So this is what syntropy means, is that just one organism and another live really close to each other. And one guy produces a waste product that the other guy happens to need for their metabolism. And so here's their relationship. Okay. All right. Thanks for watching, and I will see you in the next video.